Hello everyone. I'll be speaking about the biological control of toadflax in Colorado. Most or all of you are already familiar with this weed, so I won't go into the different aspects of the plant, other than to point out that there are two species, yellow toadflax, which occurs usually at higher elevations, and Dalmatian toadflax. Both species can outcompete native grasses and forbs on forest and rangelands. And toadflax is slightly toxic to cattle and wildlife, and so is not considered a good forage. The primary biocontrol agent released for control of toadflax is the toadflax stem weevil. There are two species, Macinus janthinus for yellow toadflax and Macinus janthiniformis for Dalmatian. They look very similar, and in fact were thought to be just one species until about 2011, when DNA analysis found that there were actually two separate species. Their life cycle is almost identical, so I'll just go through it quickly for the yellow toadflax weevil. They spend almost their entire life cycle within the stem, with the adult overwintering within. The adults emerge the following spring, mate, lay eggs within new toadflax stems. The larvae feed within the stem and cause extensive damage. They pupate and the adult continues feeding until winter. Releases were made by the Palisade insectary as early as 2009 from insects sent from the Forest Service in Montana. And since then, thousands have been released or collected from areas where we've built up populations and then re-released throughout the state. Thankfully, we continue to get weevils periodically from USDA APHIS in Montana. Of course, we can't just release biocontrol agents. It's necessary to monitor their populations to see how well they're working. For Janthinus, we have set up 12 different monitoring sites at various locations and elevations throughout the state. We work with many different cooperators at these sites. For example, Lake Avery is a state wildlife area. Burrow Mountain is on Forest Service land. Schutte Gulch is on oil and gas property. Staunton is a state park. And Windhorse is actually a private ranch. So we work with lots of different cooperators. We use two different monitoring protocols for toadflax. The first is called macro plot monitoring and consists of a large circular area of just over 2 acres, represented by the diagram on the left. The center is the initial release point of the weevil, and the white area is where we set up 16 1-meter squares and count every toadflax stem. The photo in the middle shows 2 of the 16 squares in use. We just continuously flip those over to get all 16 done. The macro plot also contains 6 transects radiating out from the release point for 50 meters every 60 degrees. Along those transects, we can determine how fast the weevils are spreading by using a nested frequency frame like the one shown in the diagram on the right, and recording toadflax frequency every 2 meters along the transect. Along with that, we record vegetation encountered along the transects to map plant community changes through time. We also use spring beetle sweeps to determine changes in the density of the weevils. Here's an example of one of the macroplot sites, the Windhorse Ranch near Sedalia, Colorado. Weevils were released here in 2013, and we can follow stem density near the release point. relative frequency of toadflax as the weevils move outward from the release point, and vegetation changes since the initial releases. As you can see, this area is trending in a good direction, even though it has taken some time. The other type of monitoring we do now is called Standardized Impact Monitoring Protocol, or SIMP for short. This protocol was developed in the Pacific Northwest by some of the folks that are probably attending this webinar. It's much faster than the macro plot monitoring, so we can set up more sites in more areas. It consists of a permanent 20-meter transect set up between two posts. A Daubenmeier frame is placed every 2 meters, and we estimate the percent cover of toadflax, other weeds, forbs and shrubs, perennial grasses, bare ground, and litter. We also count the toadflax stems found within the frame and measure and record the height of the tallest toadflax stem. After that, we conduct six 3-minute searches near the transect, counting the number of adult toadflax weevils. We also added one other component to the protocol because we were finding that we were sometimes seeing weevil damage on a lot of plants, but were not detecting any adult weevils. This was happening even in mid-June at our higher sites. We found that the adults had already laid eggs and began to die off, so we began sampling 25 nearby toadflax stems, splitting them open and looking for eggs, larvae, pupa, and adults, then recording the percent of the 25 stems that had at least one weevil in any life stage. Here's one example of a SIMP monitoring for toadflax weevil from the Flying W Ranch near Colorado Springs. This area was burned during the Waldo Canyon fire of 2012. In June of 2015, after the initial monitoring, 200 weevils were released, and the site has been monitored every year since. This graphing style was also adapted from those used by some of the SIMP protocol developers. It shows toadflax stem density on the left axis and the blue bars, while at the same time showing adult weevil density on the right axis and the black line. You'll notice that in 2017 and 18, we found no adults during the visual counts. Both of these years, the sampling was conducted in late June or very early July. It wasn't until the 2018 year that we finally figured out that we were missing weevils by not looking inside stems. So even though no adult weevils were found that year, we did find numerous immature weevils in most of the stems we sampled. After that, we started sampling 25 nearby stems and splitting them open to record weevil presence at all of our SIMP sites. This particular year, the last year, 2022, we didn't find a single toadflax stem inside the plot. We also didn't find any weevils this year either in the 25 stem sample. We are also conducting a phenology study for yellow toadflax. The goals of this study are to learn the plant and insect phenology by date, and more importantly by degree day, and to be able to determine when weevils are at their peak for collection purposes, hopefully anywhere in the West. 4 sites were set up in 2019, 2 along the Front Range of Colorado and 2 on the western slope, all at different elevations. Each site had 10 2-meter diameter permanent plots where weevils were released. Weekly monitoring starts with a 3-minute time count looking for toadflax predators, followed by selecting 20 toadflax stems at random and recording the height of each and looking for the first indication of a change in life stage. For example, at the Lake Avery State Wildlife Area, This year we found toadflax emerging on April 13th, followed by the first weevils, first flower buds, first flowers open, and then seeding and finally senescence of the weed on August 25th. This chart looks at all 4 sites by degree day for the 2022 season. As you can see, the Spruce Meadows site is a little different, but the others follow a pretty close pattern of phenology by degree day. When we look at peak numbers of weevils by degree day, so far we don't see the pattern we were hoping for when looking at the different sites. Now, the number of weevils is always going to be different at each site, with the Two Elk site near Minturn having a lot more weevils than the other sites. But the important part here is when the peaks occur. We would hope to see each line have the highest count of weevils around the same degree day. As you can see, there are sometimes several peaks, which could indicate another generation, but we don't think that's what's happening. It could be that there are just so few weevils present at some of the sites that we're not always finding what might be there if the populations were a little larger. Or perhaps there's something else going on that we will figure out as we gather more data through the years. There's a promising new toadflax predator that has just recently been approved for release in the United States. The toadflax stem gall weevil, Rhinusa pilosa, has been in use in Canada for a while now. The adults overwinter in soil and leaf litter and then emerge in the spring and lay eggs within new stems. Larvae and adults both feed within the stem, forming protective galls like those seen in the right-hand photo. Early efficacy studies showed that the gall plants were 55% shorter than controls, and most importantly, since toadflax spreads mostly through underground modified stems, galled plants had 75% less biomass below ground and also produced fewer flowering stems, as toadflax does spread by seed as well. We're hoping to receive some of these weevils this year from Montana to release here. Moving on now to the Dalmatian toadflax stem weevil, you'll see that the Palisade insectary has released hundreds of thousands of these weevils since 2003, when weevils were first obtained from the USDA APHIS. Although some of the earlier releases may have been put out on the wrong species of toadflax before we realized there were 2 separate weevil species. We have 7 different permanent monitoring sites for Janthiniformis in 3 distinct areas of Colorado. Today we'll just show some of the monitoring data from 4 of the sites located northwest of Fort Collins, Colorado. This is the location of one of our most successful biocontrol projects. It's located within the Hewlett Gulch burn of 2012. Back then, the Hewlett Gulch fire and the contiguous High Park fire burned approximately 95,000 acres in the Roosevelt National Forest. The following spring, the burn area was invaded by several noxious weeds, chief among them Dalmatian toadflax. The hillsides were covered in yellow when the toadflax flowered later in the spring. To battle these invasives, the Colorado Department of Agriculture began a cooperative effort titled the Poudre and Invasive Species Project. This project was a significant effort involving everyone that had interests in the area, from private landowners to city, county, state, and federal agencies, 10 different agencies in all. The Palisade insectary was charged with the biocontrol aspect of the project, since it was easily seen that this would be the best strategy to help control toadflax in this steep, rugged terrain. In the spring of 2013 and 14, the insectary released over 8,500 adult weevils at 27 different plots within the Hewlett Burn. We've been monitoring 4 of those plots using MacroPlot protocol annually since 2013. Things started out slowly at first, but as the weevil populations began to increase, the toadflax population began to shrink. Until in 2016, the toadflax numbers plummeted, and we found weevils on nearly every plant. In the photos, you can see how one plot looked the year after the fire. The 2016 photo was taken the same time of year at the same location. Most of the toadflax was gone by then. This chart shows the average toadflax density in the 16 1-meter squared plots for all 4 monitoring plots over the 10-year study. You can see the dramatic decrease that took place in 2016 and that the toadflax numbers have stayed relatively low since then. The weevil numbers found by sampling overwintering stems in the fall have remained fairly constant at a high level. The Toadflax program probably wouldn't exist without funding assistance from the USFS and APHIS. So thanks to those folks. And also I want to extend a special thanks to all of you, because whether you helped to get these biocontrol agents approved, provided us with biocontrol agents, assisted in release and monitoring, or allowed access to locations to conduct these studies, you are the ones that make biocontrol work. I also want to thank all my colleagues here at the Insectary, including the many seasonal employees we've had through the years.